Hey guys, welcome to our animation on quorum sensing. In essence, quorum sensing is basically bacterial gossip, so you have one bacteria talk to another.
Quorum sensing is a process that allows bacteria to produce signals that cause cell-dependent phenotypes. This process is regulated by the production of signaling molecules called autoinducers. Quorum sensing is used by gram-negative bacteria where they produce a specific class of signals called AHLs. These AHLs are secreted and result in gene expression and phenotypes like plant growth and bioluminescence. And we learned that plant growth is optimal, with a low concentration of AHLs and acyl chain length of around 12 carbons. And plant growth doesn't even depend on the entire AHL molecule, but rather the amyloidosis product L-homoserine. So AHLs, after being cleaved to L-homoserine, increase plant growth by increasing transpiration, which is water loss that increases mineral and water uptake in the roots that causes overall plant growth.
These quorum sensing signals affect plant growth, but not on their own. Plants that grow in soil with AHLs present have an enzyme called fatty acid amide hydrolase, or FAAH, in their roots. Plants can eavesdrop on the AHL signals produced by bacteria by using the FAAH enzyme to hydrolyze the signal into a molecule, L-homoserine, that will ultimately induce plant growth through transpiration.
We saw how quorum sensing affects plants, so now we'll go through a basic mechanism of quorum sensing to relate it to both plants and animals. As we now know, quorum sensing is regulated by production of AHLs. So we have our bacteria, and inside is its cell that contains a protein called AHL synthase that creates AHLs. Those AHLs are then secreted out of the cell, and at high concentrations, they are brought back into the cell. Once inside again, the AHL binds to a quorum-sensing receptor, which also binds to DNA to initiate transcription and ultimately leading to gene expression. Animals, like the bobtail squid, use quorum sensing to produce bioluminescence. When there is a low cell density, there is no luminescence, but at high cell density, as stated earlier, we see luminescence.
And that's all, folks.